A soldier lies dying in a North African field hospital, not from his wounds, from what came after. Blood soaked through the bandages faster than the medics could replace them. The young private's leg wound wasn't even that bad. A clean shot through the calf muscle, no major arteries hit. 3 days ago, he would have walked out of this tent hospital with a limp and a story to tell. But this was 1942, and the real enemy had already invaded his bloodstream. The infection spread like wildfire through damaged tissue. His temperature spiked to 105 degrees. Delirium set in. The medics knew what was coming next. Sepsis, organ failure, death. They had morphine to ease his pain and sulfa drugs that barely slowed the bacterial onslaught. They had bandages and surgical tools and blood plasma. They had everything except the one thing that could actually save him. This scene repeated itself thousands of times across every theater of World War II. In the jungles of Guadalcanal, where the humidity turned every scratch into a festering wound. On the beaches of Sicily, where shrapnel embedded dirt and fabric deep into flesh. In the frozen forests of the Eastern Front, where frostbite opened doors for gangrene. The statistics told a grim story. 18% of wounded Allied soldiers died not from their injuries, but from the infections that followed. 18%. Nearly 1 in 5. Bullets and bombs got the headlines. Bacteria did the killing. Military planners understood the mathematics of war. They calculated ammunition expenditure per enemy casualty, fuel consumption per mile advanced, replacement rates for destroyed equipment. But infection was a variable they couldn't control, a force multiplier for the enemy that operated invisibly on their own side. Every soldier pulled from the line by infected wounds meant one less rifle on the front, one more burden on supply lines, one more letter home that began with, "We regret to inform you." The problem had plagued armies throughout history. More soldiers died from disease and infection than from combat in virtually every war before the 20th century. The American Civil War saw twice as many deaths from disease as from battle. World War I introduced modern weapons that created horrific wounds, but medical science had no answer for the infections that followed. Field hospitals became warehouses of suffering, places where young men went to die slowly from wounds that should have been survivable. Military doctors tried everything. They debrided wounds, cutting away dead tissue. They used antiseptics that burned like fire. They amputated limbs at the first sign of gangrene. They isolated infected patients to prevent spread. Nothing worked consistently. Bacteria were invisible enemies that couldn't be shot, bombed, or outmaneuvered. They simply multiplied and killed. The British had discovered something that could change this equation. Back in 1928, a Scottish bacteriologist named Alexander Fleming had noticed something odd in his laboratory at St. Mary's Hospital in London. A petri dish contaminated with mold had killed all the bacteria growing around it. He identified the mold as Penicillium notatum and named its antibacterial substance penicillin. Fleming published his findings, but the discovery languished for over a decade. Nobody could figure out how to produce penicillin in meaningful quantities. The substance was maddeningly difficult to extract, unstable, and required enormous amounts of mold culture to generate even tiny quantities of usable medicine. Fleming himself was pessimistic about penicillin's practical applications. The mold grew slowly. The antibiotic it produced was present in minuscule concentrations. Extracting and purifying it required complex chemical procedures. Even under ideal laboratory conditions, researchers could barely produce enough penicillin to treat a single patient. The drug would degrade before it could be stored or transported. It seemed like a laboratory curiosity, scientifically interesting, but medically useless. Then the war came, and suddenly those tiny quantities looked like liquid gold. By 1939, a team at Oxford University led by Howard Florey and Ernst Boris Chain had revived interest in penicillin. They improved extraction methods and conducted the first clinical trials. The results were miraculous. A policeman dying from a bacterial infection improved dramatically after receiving penicillin. Unfortunately, the Oxford team had so little of the drug that they collected the patient's urine, extracted unmetabolized penicillin from it, and reused it. When their supply ran out, the patient relapsed and died. But those few days of improvement proved the concept. Penicillin could save lives if only they could produce enough of it. By 1941, British researchers had successfully treated several patients with penicillin. Infections that would have meant amputation or death simply vanished. Soldiers returned to duty within days instead of weeks. The problem was scale. British laboratories were producing penicillin by growing mold in hundreds of shallow ceramic vessels, like a bizarre dairy operation. The entire British supply couldn't treat more than a handful of patients. With German bombs falling on London and resources stretched to breaking point, the British couldn't scale up production fast enough. Florey traveled to the United States in 1941 to seek American help. He brought samples of Penicillium notatum and data from the Oxford trials. The timing was perfect. America was gearing up for war, mobilizing its massive industrial capacity. The government understood that medical innovation could be as important as weapons development. If penicillin could keep soldiers fighting, it was worth any investment. The Americans inherited the problem. In 1941, the U.S. government launched a massive effort to crack penicillin production. The challenge landed at the Northern Regional Research Laboratory in Peoria, Illinois, an unlikely epicenter for a medical revolution. The NRRL was a Department of Agriculture facility that normally studied corn processing and fermentation, but it had something crucial: expertise in growing microorganisms in large quantities. Peoria was chosen for practical reasons. The laboratory had experience with industrial fermentation. The region produced vast quantities of corn, and corn processing generated waste products that might be useful for growing mold. The facility had space, equipment, and scientists who understood how to scale up biological processes. It wasn't glamorous, but it was practical. The scientists at Peoria understood the core problem. Fleming's original Penicillium notatum strain was a poor producer. Even under optimal conditions, it generated barely enough penicillin to treat a single patient from liters of culture. The British shallow pan method was labor-intensive and inefficient. What they needed was a better strain, a genetic variant that could pump out penicillin faster and in greater quantities, and a better production method that could scale to industrial levels. Somewhere in the world, such a strain might exist. The question was where. Penicillium molds are everywhere. They're the blue-green fuzz on forgotten bread, the dusty coating on aging cheese, the spots on overripe fruit. Each colony represents a unique genetic lineage, shaped by countless generations of mutation and selection. Most produce little or no penicillin, but statistical probability suggested that somewhere, some strain had evolved the genetic machinery to produce the antibiotic in abundance. The genetics were pure chance. Penicillin production requires a complex biochemical pathway involving multiple enzymes. Mutations that enhance any step in that pathway could increase output. Mutations that create more copies of key genes could multiply production. In the vast global population of Penicillium molds, spanning millions of colonies across every continent, the odds suggested that high-producing strains existed. Finding them was the challenge. The NRRL launched a systematic search. They put out a call to laboratories, universities, and military personnel around the world. Send us your moldy samples. Packages arrived from every continent: soil from battlefields in North Africa, fruit from tropical islands in the Pacific, cheese from caves in France, bread from bakeries in South America. Each sample went through the same rigorous testing protocol: isolate the mold, grow it in culture, extract any penicillin produced, measure the yield. Hundreds of samples. Hundreds of failures. Most samples contained no penicillium at all, just other molds or bacteria. Of those that did contain penicillium, most produced no penicillin. Of those that produced penicillin, most generated amounts similar to or less than Fleming's original strain. The search was tedious, expensive, and mostly fruitless, but the scientists persisted because the alternative was unacceptable. Without better penicillin production, thousands of soldiers would die from preventable infections. The scientists needed more samples faster. They needed someone to actively hunt for promising molds in the local area, someone who could collect specimens daily and deliver them fresh to the laboratory. The job didn't require an advanced degree or specialized training. It required patience, attention to detail, and a willingness to dig through garbage, looking for rot. They hired Mary Hunt. Mary Hunt was a laboratory technician, not a scientist. She had no doctorate, no published research, no academic reputation. She was exactly the kind of person who fades into the background of history, doing necessary work that nobody remembers. Her job description was simple and strange: Go to markets, grocery stores, and anywhere else food was sold or discarded. Look for mold. Bring back samples. Her colleagues started calling her Moldy Mary. The nickname was affectionate but revealing. In a laboratory full of PhDs and serious researchers working on a project that could change the course of the war, Mary Hunt was the woman who collected garbage. The hierarchy was clear. The scientists did important work. Mary Hunt did the dirty work that supported them. She didn't complain. She understood the mission. Every sample she collected was a lottery ticket. The odds of any individual sample containing a miracle strain were tiny, but someone had to buy the tickets. That someone was her. Every day, Mary Hunt walked through Peoria with paper bags and collection containers. She visited produce markets where vendors tossed out unsellable fruit. She checked bakery dumpsters for moldy bread. She examined cheese shops and asked grocers if they had anything spoiled in the back. The work was unglamorous, repetitive, and often disgusting. Rotting food smells like death for a reason. It's organic matter breaking down, returning to the chemical soup from which it came. Most people avoided moldy food instinctively. Mary Hunt sought it out deliberately. She developed an eye for promising specimens. Not all mold was penicillium, and not all penicillium looked the same. She learned to recognize the characteristic blue-green color, the dusty texture, the particular way it spread across organic matter. She learned which substrates were most likely to harbor interesting strains. Citrus fruits often had good molds. Melon sometimes. Bread was hit or miss. Cheese was usually disappointing because the molds were intentionally cultivated varieties. She labeled each sample carefully. Location found, type of substrate, date collected, any notable characteristics. Then she delivered them to the laboratory where scientists would begin the tedious process of isolation and testing. Most samples went nowhere. The mold would be the wrong species, or it would produce no penicillin, or it would produce trivial amounts. Mary would note the result and move on to the next sample. The scientists were looking for a needle in a haystack, except they didn't know what the needle looked like or even if it existed. Each sample represented hope, and almost always, disappointment. This strain produced slightly more penicillin than Fleming's original. That strain produced slightly less. None produced enough to matter. The war ground on. Casualties mounted. The pressure intensified. By mid-1943, the Peoria laboratory had tested over 1,000 samples from around the world. They had found a few strains that were marginally better than Fleming's original, but nothing that would enable true mass production. The pharmaceutical companies were gearing up to produce penicillin using whatever strains were available, but everyone knew the yields would be disappointing. The military would get some penicillin, enough to treat the most critical cases, but not enough to make a real difference in overall casualty rates. The clock was ticking. Military planners were preparing for the invasion of Europe. The date was secret, but everyone knew it was coming in 1944. The invasion would produce casualties on a scale not seen since World War I. Tens of thousands of wounded soldiers would need immediate medical care. Without adequate penicillin supplies, many would die from infections. The success of the invasion might depend partly on whether wounded soldiers could be kept alive and returned to duty quickly. Summer 1943. Mary Hunt had been collecting samples for months. The routine had become automatic. Walk the markets, examine the discards, collect anything promising, deliver to the lab. Most days blurred together. This particular day seemed no different. She was at a fruit market in downtown Peoria. The summer heat made the work even less pleasant. Rotting fruit in August smells worse than rotting fruit in cooler months. The vendor was sorting through crates of cantaloupes, pulling out the ones too ripe to sell. One melon caught her eye. It sat at the bottom of a discard bin, its rind covered in a thick coating of golden green mold. The mold had that characteristic dusty appearance of penicillium, but the color was unusual, more golden than the typical blue-green. The melon was thoroughly rotten, its flesh collapsed and liquefied inside the rind. Mary Hunt almost passed it by. She had collected dozens of moldy melons before. None had yielded anything interesting, but something about this one made her pause. Maybe it was the unusual color. Maybe it was intuition. Maybe it was just thoroughness. She placed the disgusting melon in her collection bag and continued her rounds. The melon rode back to the laboratory in her bag, its rotten smell mixing with the other samples she had collected that day. Just another specimen in an endless series of specimens. just another lottery ticket that would probably lose. Back at the laboratory, the Melon joined the queue of samples awaiting processing. A technician scraped mold from the rind and transferred it to a sterile growth medium. The culture grew overnight. The next day, scientists transferred the mold to a fermentation flask containing corn steep liquor, a waste product from corn processing that Peoria researchers had discovered made excellent mold food. The flask went into an incubator. Days passed. The mold grew, spreading through the liquid medium and turning it cloudy. When the culture reached peak growth, technicians extracted the liquid and tested it for penicillin content. This was the moment of truth for every sample. Most produced nothing. Some produced trace amounts. Very few produced enough to merit further investigation. The numbers didn't make sense at first. They ran the test again. Same result. They brought in senior researchers. Everyone stared at the data. This strain was producing penicillin at levels nobody had seen before. Not slightly more than previous strains. Not incrementally better. This mold was generating 200 times more penicillin than Fleming's original Penicillium notatum. 200 times. The scientists ran more tests to confirm. They grew the mold in different media. They tested it under different conditions. They verified the penicillin was genuine and not some testing artifact. Every test confirmed the same conclusion. This was the strain they had been searching for. The scientists cataloged the strain as NRRL 1951. Its scientific name was Penicillium chrysogenum, golden penicillium, named for its distinctive color. It had been growing on a rotten cantaloupe in a Peoria market, waiting for someone to notice it. If Mary Hunt had visited that market a day later, the melon would have been gone. If she had decided to skip that location, someone else would have thrown it away. If she had glanced at it and decided she had enough melon samples already, it would have rotted in a dumpster. Instead, she collected it, and that decision changed everything. Mary Hunt had found the needle in the haystack. The discovery triggered a cascade of activity. The NRRL immediately began scaling up production of the new strain, but even a super-producing mold wasn't enough. The British method of growing penicillium in shallow trays wouldn't work for mass production. Growing mold in thousands of shallow pans would require enormous amounts of space, labor, and materials. It was fine for laboratory-scale production, but impossible for the industrial scale needed to supply a global war. The Peoria team had been developing an alternative, deep tank fermentation, the same technology used to brew beer on an industrial scale. Instead of growing mold on the surface of shallow liquid, they would grow it throughout the entire volume of large tanks. The process worked like this. Massive steel tanks, thousands of gallons in capacity, filled with corn-steep liquor and other nutrients. Sterile air pumped through the liquid to provide oxygen. Temperature carefully controlled. The NRRL 1951 strain inoculated into this environment and left to grow. The mold didn't just sit on the surface like in shallow trays. It grew throughout the entire volume of liquid, billions of microscopic filaments churning out penicillin. The engineering challenges were substantial. The tanks had to be completely sterile before inoculation. Any contaminating bacteria or other molds would ruin the batch. The air pumped through had to be filtered to remove contaminants. The temperature had to be maintained precisely. The nutrient mixture had to be optimized for maximum penicillin production. Every variable mattered. But the Peoria team solved the problems one by one. They developed sterilization procedures. They designed filtration systems. They optimized the growth medium. By late 1943, they had a working process that could produce penicillin in quantities that would have seemed impossible a year earlier. The pharmaceutical industry mobilized. Pfizer, Merck, Squibb, and other companies converted facilities to penicillin production. Engineers designed specialized fermentation tanks based on the Peoria specifications. Chemists optimized extraction and purification methods. The government provided unlimited funding. This was a wartime crash program like the Manhattan Project, but aimed at saving lives instead of taking them. Pfizer took a particularly bold approach. The company had experience with fermentation technology from producing citric acid. They converted an entire factory in Brooklyn to penicillin production, installing massive fermentation tanks and extraction equipment. It was a huge financial risk. If the process didn't work at industrial scale, Pfizer would lose millions. But the company's leadership understood the stakes. This wasn't just about profit. This was about winning the war. The production numbers told the story. In 1942, before Mary Hunt's Melon, total U.S. penicillin production measured in grams, enough to treat a few dozen patients. By early 1943, production reached hundreds of grams per month. By late 1943, after NRRL 1951 went into mass production, output jumped to kilograms. By early 1944, pharmaceutical companies were producing penicillin by the ton. The scale-up was one of the most impressive industrial achievements of the war. In less than 2 years, penicillin went from a laboratory curiosity to a mass-produced medicine. The transformation required breakthroughs in microbiology, chemical engineering, industrial fermentation, and manufacturing. It required cooperation between government laboratories, universities, and private companies. It required massive investment with no guarantee of success, and it all depended on a mold from a rotten cantaloupe. The military had a deadline, June 1944. Everyone knew the invasion of Europe was coming. The exact date was the most closely guarded secret of the war, but the medical corps knew they needed to be ready. An amphibious assault on fortified beaches would produce casualties on a scale not seen since World War I. Thousands of wounded men would need immediate treatment. Infection had to be controlled, or the invasion could fail from sheer medical logistics. Military medical planners studied the problem carefully. They estimated casualty rates based on previous amphibious operations. They calculated how much penicillin would be needed per wounded soldier. They determined how much could be transported and stored in field conditions. They trained medics on proper dosage and administration. Every detail mattered. By June 1944, the stockpiles were ready. Field hospitals received shipments of crystalline penicillin in sealed vials. Medics received training on dosage and administration. For the first time in military history, frontline medical personnel had a weapon that could stop infection before it started. The drug that had been rarer than gold in 1942 was now available in quantities sufficient to treat an entire invasion force. June 6, 1944. D-Day. Allied forces hit the beaches of Normandy. The casualties were as bad as predicted. Over 10,000 Allied soldiers killed or wounded on the first day alone. Medics worked frantically in aid stations just behind the front lines, treating gunshot wounds, shrapnel injuries, burns, fractures. And now they had penicillin. A soldier hit by machine gun fire, bullet fragments driven deep into muscle tissue along with cloth and dirt. In previous wars, this wound would almost certainly become infected. The bacteria from the cloth and dirt would multiply in the damaged tissue. Within days, the wound would be red, swollen, and oozing pus. Fever would set in. Without effective treatment, the infection would spread through the bloodstream. Sepsis, organ failure, death, or if the soldier was lucky, amputation of the affected limb before the infection spread too far. Now the medic cleaned the wound as best he could under battlefield conditions. He injected penicillin directly into the tissue around the wound. He bandaged the soldier and sent him back to a field hospital. There, surgeons would remove the bullet fragments and dead tissue. More penicillin would be administered. The soldier would survive. The wound would heal. Within weeks, he would return to his unit. Another soldier, legs shredded by artillery shrapnel, multiple penetrating wounds. Each piece of shrapnel carried dirt and debris deep into tissue. Each wound was a potential site of infection. In World War I, this soldier would have died or lost both legs. Now, penicillin, survival, recovery, eventually, walking again. A tank crewman, severe burns from a vehicle fire, skin barrier destroyed and vulnerable to every bacterium in the environment. Burns were particularly prone to infection because the protective skin layer was gone. Bacteria could invade directly into underlying tissue. Before penicillin, severe burns often meant death from infection, even if the patient survived the initial injury. Now, penicillin, survival, skin grafts, eventually, home. The statistics shifted dramatically. In World War I, 18% of wounded soldiers died from infection. In the early years of World War II, before widespread penicillin use, the rate was similar. After D-Day, with penicillin available at the front lines, the infection mortality rate dropped to 1 to 2%. 1 to 2%. Penicillin didn't just save lives, it changed the fundamental mathematics of combat medicine. The impact extended beyond Europe. In the Pacific Theater, where tropical diseases and jungle conditions made infection even more likely. Penicillin proved equally revolutionary. Island-hopping campaigns produced horrific wounds. Japanese defenders fought to the death. Battles were close quarters and brutal. The coral and volcanic rock of Pacific Islands ground into wounds, carrying bacteria deep into tissue. Before penicillin, these wounds festered in the heat and humidity. With penicillin, soldiers survived injuries that would have killed them months earlier. The Battle of Iwo Jima in February 1945 demonstrated penicillin's impact. The battle produced over 26,000 American casualties in just over a month of fighting. The volcanic ash of Iwo Jima contaminated every wound. In previous wars, the infection rate would have been catastrophic. With penicillin, the vast majority of wounded soldiers survived. Medics administered the drug in the most hellish conditions imaginable. Under fire, in foxholes, on beaches swept by artillery. The wounded survived long enough to reach hospital ships offshore. The hospital ships had more penicillin. Survival rates stayed high even in the worst conditions. The Battle of the Bulge, winter 1944-45. German forces launched a massive surprise offensive through the Ardennes forest. American units were surrounded, cut off, fighting in brutal cold. Wounded men lay in frozen foxholes for hours before evacuation. Frostbite complicated every injury. In previous wars, these conditions would have meant death sentences from gangrene and infection. Penicillin changed the equation. Medics administered the drug in field conditions, sometimes with frozen fingers, sometimes under fire. The wounded survived long enough to reach field hospitals. The field hospitals had more penicillin. Survival rates stayed high even under the worst conditions. By 1945, penicillin production had become a massive industrial operation. American pharmaceutical companies were producing 650 billion units per month, enough to treat hundreds of thousands of patients. The drug that had been rarer than gold in 1942 was now available to every military hospital and aid station. Civilian doctors began receiving supplies. The antibiotic revolution had begun. Mary Hunt continued working at the NRRL throughout the war. She kept collecting samples, kept hunting for molds, kept doing the unglamorous work that nobody noticed. She received no promotion, no medal, no public recognition. Her name didn't appear in newspaper headlines or military dispatches. While generals received decorations for victories that penicillin had helped make possible, Mary Hunt remained a laboratory technician in Peoria, Illinois. The contrast is almost absurd. A cantaloupe from a market discard bin contributed more to Allied victory than many decorated officers. A woman walking through grocery stores with paper bags saved more lives than most combat medics. The invisible infrastructure of war, the laboratories, the factories, the supply chains, the ordinary people doing extraordinary work. These determined outcomes as much as battlefield tactics. Consider the chain of causation. One rotten melon, collected by one persistent technician, yielded one exceptional mold strain. That strain enabled mass production of one antibiotic. That antibiotic saved thousands of lives directly and enabled military operations that saved thousands more indirectly. The invasion of Europe succeeded partly because Allied commanders knew their wounded would survive. Soldiers fought more aggressively knowing infection wasn't a death sentence. The entire strategic calculus of the war shifted because of a fungus growing on discarded fruit. The numbers tell part of the story. Estimates suggest penicillin saved between 12 and 15% of wounded Allied soldiers who would have died from infection in previous wars. Across all theaters of World War II, that translates to somewhere between 100,000 and 200,000 lives saved. 100,000 to 200,000 soldiers who went home instead of dying in field hospitals. 100,000 to 200,000 families who didn't receive telegrams beginning with, "We regret to inform you." But numbers can't capture the full impact. Every soldier saved by penicillin had a life after the war. They had children who wouldn't have been born. Those children had children. The descendants of penicillin-saved soldiers now number in the millions. A rotten melon in 1943 has a family tree that spans generations. The scientific legacy is equally profound. NRRL 1951 became the foundation of the modern penicillin industry. Pharmaceutical companies subjected the strain to further mutation and selection, creating even more productive variants. They exposed the mold to X-rays and ultraviolet light to induce mutations. They tested thousands of mutant strains to find ones that produced even more penicillin. By the 1950s, they had strains that produced 1,000 times more penicillin than Fleming's original. But the genetic lineage traced back to Mary Hunt's cantaloupe. Every dose of penicillin produced today descends from that single moldy fruit. The discovery also validated a new approach to drug development. Before penicillin, most medicines were either extracted from plants or synthesized chemically. The idea of systematically searching for microorganisms that produced useful compounds was novel. After penicillin's success, pharmaceutical companies launched massive screening programs. They collected soil samples from around the world, isolated microorganisms, and tested them for antibiotic activity. This approach yielded streptomycin, discovered in soil bacteria in 1943. Streptomycin was effective against tuberculosis, a disease that penicillin couldn't treat. Then came tetracycline, erythromycin, chloramphenicol, and dozens of other antibiotics. Each discovery came from screening thousands of microbial samples. The entire modern antibiotic arsenal traces its development strategy back to the penicillin program. The impact on medicine was revolutionary. Before antibiotics, bacterial infections were major killers. Pneumonia, tuberculosis, infected wounds, childbed fever, bacterial meningitis. These diseases killed millions every year. Antibiotics transformed them from death sentences to treatable conditions. Life expectancy increased dramatically in the decades after World War II, partly because bacterial infections became manageable. Mary Hunt's story eventually emerged from obscurity, but it took decades. In the immediate post-war years, credit for penicillin went to the obvious names: Fleming for discovery, Florey and Chain for development, the pharmaceutical companies for production. The supporting cast, the technicians, the sample collectors, the factory workers, faded into historical background noise. Fleming, Florey, and Chain shared the Nobel Prize in Physiology or Medicine in 1945. The award was deserved. Their work was groundbreaking. But the Nobel Prize can only go to 3 people, and it tends to recognize the lead scientists rather than the supporting team. Mary Hunt was never in consideration. Her contribution, while crucial, was seen as technical rather than scientific. She collected samples. She didn't design experiments or analyze data or publish papers. Gradually, historians began documenting the full story. They interviewed surviving participants, examined laboratory records, pieced together the details of the Peoria program. Mary Hunt's name appeared in academic papers and historical accounts. Her contribution was recognized, though never with the prominence given to the lead scientists. She lived a quiet life after the war. No fame, no fortune, just the satisfaction of work well done. When she died, her obituary mentioned her role in the penicillin program, but most people reading it probably didn't grasp the significance. How do you compress saved hundreds of thousands of lives into a newspaper paragraph? The broader lesson extends beyond one person or one discovery. World War II was won by logistics as much as tactics, by industrial capacity as much as military skill. The Allies prevailed because they could produce more tanks, more planes, more ships, more ammunition than the Axis powers. But they also prevailed because they could keep their soldiers alive. Medical innovation was a force multiplier that doesn't appear in traditional military histories. Penicillin was part of a larger pattern. Blood plasma and improved transfusion techniques reduced deaths from hemorrhage. Better surgical methods saved soldiers who would have died on operating tables in previous wars. Anti-malarial drugs kept troops fighting in tropical regions. Improved field sanitation reduced disease casualties. DDT controlled typhus-carrying lice. Each innovation came from unglamorous research, often conducted far from battlefields by people whose names never made headlines. The pattern continues today. Modern military medicine builds on the foundation laid during World War II. Antibiotics are standard in every medical kit. Combat medics carry drugs that can stop bleeding, prevent infection, and stabilize patients for evacuation. Survival rates for wounded soldiers are higher than ever, not because wounds are less severe, but because medical science has advanced. And it all traces back, in part, to a woman walking through Peoria markets with paper bags, looking for rotten fruit. There's something profound in that image. The contrast between the monumental and the mundane. A global war involving millions of soldiers, Thousands of tanks and planes, entire nations mobilized for total conflict. And in the middle of it all, a laboratory technician examining moldy cantaloupes. The juxtaposition seems almost comical until you follow the consequences. History tends to remember the dramatic moments. The beach landings, the tank battles, the air raids, the generals making strategic decisions. These are the scenes that make it into textbooks and movies. The supporting infrastructure, the supply clerks, the factory workers, the researchers, the technicians, these fade into the background despite being equally essential. Mary Hunt represents all of them. Every person who contributed to victory without firing a shot. Every worker who built the planes and ships. Every farmer who grew the food. Every scientist who solved technical problems. Every technician who did the tedious, repetitive work that made breakthroughs possible. The invisible army that made the visible army function. The melon itself is worth contemplating. It was garbage, literally. Unsellable produce destined for the trash or compost heap. In any other context, it would have rotted completely and returned to soil, its existence noted by nobody. Instead, it became one of the most consequential pieces of fruit in human history. The mold growing on its rind saved more lives than most medical interventions ever developed. There's a randomness to it that's almost unsettling. What if Mary Hunt had visited that market a day earlier or later? What if the vendor had already thrown out that particular bin? What if Mary had been sick that day, or decided to skip that location? The NRRL would have kept searching, would have eventually found another high-producing strain. But how much longer would it have taken? How many soldiers would have died in the interim? History pivots on small moments more often than we acknowledge. The decisions that seem insignificant at the time, which market to visit, which sample to collect, can cascade into world-changing consequences. Mary Hunt couldn't have known that one particular melon would matter. She collected it because she was thorough, because she was doing her job properly, because she understood that every sample was potentially important. That's the real lesson. Heroism isn't always dramatic. Sometimes it's just showing up every day and doing the work, even when the work is unglamorous and the results are uncertain. Sometimes it's walking through markets with paper bags, examining garbage, collecting samples that will probably amount to nothing. Sometimes it's being the person who doesn't give up, who checks one more location, who brings back one more specimen. The soldier in the North African field hospital, the one dying from infection at the beginning of this story, he represents thousands of real soldiers who faced the same fate in 1942 and early 1943. But by 1944, that soldier would have survived. The medic would have injected penicillin. The infection would have cleared. He would have recovered, returned home, lived a full life. The difference between those two outcomes, death in 1942, survival in 1944, was measured in many factors. Strategic decisions, industrial production, scientific research, medical training. But it was also measured in paper bags full of moldy fruit, collected by a woman whose name most people have never heard. Mary Hunt didn't storm beaches or fly bombing missions. She didn't command armies or plan invasions. She walked through Peoria with collection bags and brought back samples. That was her contribution to victory. That was how she saved lives. Sometimes the difference between victory and defeat, between life and death, between historical triumph and tragedy, comes down to someone willing to examine rotten fruit and see possibility instead of garbage. Sometimes the hero of the story is the person nobody remembers, doing work nobody notices, making discoveries that change everything. Sometimes that hero carries paper bags through grocery stores, hunting for mold. And sometimes, just sometimes, she finds a golden green fungus on a disgusting rotten melon. And that fungus floods military hospitals with life-saving medicine. And thousands of soldiers survive who would have died. And the course of history shifts in ways that ripple forward through generations. That's the story of Mary Hunt. That's the story of NRRL 1951. That's the story of how one grocery girl's persistence and one rotten cantaloupe helped win World War II. The back lines saved the front lines. The invisible made the visible possible. And a moldy melon from a Peoria market became one of the most important medical discoveries in human history. If this story of how a rotten melon changed the course of history amazed you, imagine what other untold stories are waiting to be discovered. Subscribe to this channel so you never miss another incredible piece of history that textbooks forgot to mention. Hit that like button if Mary Hunt deserves more recognition than she ever got. And drop a comment below telling us, what other unsung heroes from history do you think we should cover next? Your engagement helps us bring these forgotten stories back to life. See you in the next one.